Everybody, I am Mr. Fizz, Dr. Fizz's doppelganger, and let's learn some physiology. So today we're going to study capillaries and blood pressure. Obviously, I am actually his stunt double, not his doppelganger. I apologize. All right, so this is my plan to dominate the world. Today we're going to talk about various forces behind movement of fluids and capillaries. We're going to talk about the use of inflammation to describe edema. We'll talk about baroreceptor reflex, give an example, and talk about regulation of blood flow. And then we'll review what we talk about. Let's get right into it. Alrighty, so various forces behind movement of fluids and capillaries. So before I talked about anything, I wanted to give a structure, kind of give us a better understanding of what a an artery will look like. Um, Veins and arterioles are just basically versions of these that are stripped down without as much smooth muscle. The benefit in this structure, we have the tunica aventia. I think that's what it says. I can't read from here. I'm sorry. And then we have the tunica lamina, as well as the tunica intima. And basically, these three sections can be broken down into further places. You can see here that we have blood vessels and nerves that go throughout this, and they connect all of these and innervate them. We have lamina that cover different levels of smooth muscles. And on the inside, we have endothelium. So all these muscles help to contract and move the blood forward through the arteries. And as time goes on, or as they go further down the body, they tend to split off and lose a lot of these layers. So along, we're going to be talking about uh, we'll be talking about different veins today too. So I wanted to talk about capillaries and veins. And I wanted to talk about different types of capillaries. So we have three different types of capillaries that we're going to cover today. One is a continuous capillary, and basically this one has no gaps whatsoever. Kind of allows for all the blood to flow through. It's endothelial, and it's impermeable to most polar molecules. So these are found in the kidneys, the nervous system, muscle, and fat, and in the heart, along with other places. Next, we have fenestrated. I never have known how to say that. Fenestrated, I think, is what it is. These are like just like continuous, except they have pores all throughout them, which allow for bigger molecules to go in and out. That just allows for. We'll talk about what capillaries do, but this will be important. All right, and then you have sinusoidal capillaries, and these are a special type of capillary in the bone marrow and the liver and the spleen. And they have large groups, uh, large gaps right here, for example, between different sections. And he's let in even bigger molecules. Alrighty, talking about capillaries and veins and how everything goes. Let's talk about the various forces behind the movements of capillaries in this way. I have a lot of things built here, so let's dissect it. I'll point out. You probably have to zoom in, but this is a capillary right here. You can obviously see that these are both two different capillaries, but they're different. One is a precapillary area where it has relaxed sphincters, which allow for blood from the arteriole going up here to actually enter into this and pick up different waste products and whatnot from the interstitial space. Here's what's called, yeah, the interstitial space. That'll get oxygenated or deoxygenated, I should say. Sorry, oxygen will go out here and interchange with waste products and then go back into the veins. Not a lot of pressure here. Here, I drew it as black, showing like there was stale blood here. I don't really know. But these are precapillary constricted sphincters. Both of these can perform autoregulation in order to allow for if there are a lot more waste products, they'll open up to let things flow through and out of this arteriole and whatnot in order to get back into the veins to be uh, pushed through the cleaning systems, you could say. Um, so this has to deal with hydrostatic pressure and IFP, which we'll talk about more in the bit. Long story short, though, let's zoom in right here and go into this area. It's kind of hard to see, but I just drew the arterial and the venous side of capillary exchange when it comes to lymphocytes. So really, we'll have like a lymph duct up here, and this is determining what goes in and what comes out of this area in order to put waste products into the lymph system. Long story short, we have pressure going out, pressure going in, i.e. net or hydrostatic and osmotic pressure. And I abbreviated it down here, HP, pressure, hydrostatic pressure, and then you have IFP, BCOP, and then ICOP, going in that order right here. So HP goes in, IFP goes in, or sorry, HP goes out, in, and out. So IFP, BCOP go in, ICOP go out. This ends up, what goes out usually attributes to net hydrostatic pressure. What goes in usually attributes to net osmotic. And right here, sorry, that's not correct actually. IFP and HP go or add to hydrostatic, and BCOP and ICOP contribute to osmotic. So you can look at that on the venous side too. Basically, all of these will come out to be a net filtration, and depending on what's outside in the interstitial space or what's inside in the capillaries, it'll exchange in order to put waste into the pretended. I'll just draw it. The pretended lymph duct right here, and it'll get cycled out of the bloodstream. So, all right. So, use of inflammation to describe edema. So this is going to be, when I say use of inflammation, we're just going to describe, we're going to use that as an example to describe edema. Abnormal accumulation of fluid in the interstitial space is the definition of edema. Basically, going back and looking at that capillary chart, I can't do that for some reason in PowerPoint, but if you have too much buildup in that space, this is called edema. Inflammation can actually cause this with the amount of proteins that are released and whatnot. By inflammation, that'll cause clotting of blood or other molecules and build up fluid in that space. And this can be caused by 4 different other things. So increased hydrostatic pressure is one of them. Increased permeability of the cells to let things out. Blockage or breakdown of the lymphatic system. So there's actually worms that will eat the lymphatic system in some people. They've found that there are worms that do that. So don't contract those because your lymph system will be destroyed. That will create no way to get any kind of extra waste product out. And there will just be a bunch of waste product in the interstitial space that the blood can't pick up. Kind of scary. And then osmotic edema has to do with the dilution of plasma. Basically, you put more water into your plasma, which causes the concentration of your solute to decrease. Even though there's the same amount of solute, concentration would decrease. And so that will cause more fluid in the interstitial space, i.e. edema, and cause inflammation because of that and swelling. All right, now my favorite part. We're going to talk about the baroreceptor reflex. The baroreceptor reflex is basically a mechanically, excuse me. Sorry, I just took a test. Anyway, baroreceptors are mechanically gated. So when they receive pressure, they will send signals up to the medulla, which controls the heart rate and the force of contraction and the diameter of arterioles. So that is the purpose of medulla, but it'll send it through cranial nerve 9. Let's see. So when there's more stretch, i.e. there's more pressure put on a baroreceptor, that means there's more signal. There's one in the aorta, and there's one. Would I say superficial to it? No, not superficial. I would say superior to it. Yeah, superior to the aorta. Um, so when there's pressure put on it, it'll send that signal to the cranial nerve, and then the cranial nerve will tell the medulla, the control center, hey, this is going wrong. So it'll send a parasympathetic or a symp or an sorry, it'll send a parasympathetic or a sympathetic response to that, depending on what's happening. Generally, if there's not enough pressure, it'll send probably a sympathetic system and cause it to beat more. If it's sends a parasympathetic, it will cause it to relax a little more. Flight or flight response. Alrighty, moving on. Something that the medulla can also do is send signals to the adrenal medulla on the kidney, which will produce epinephrine and norepinephrine. Norepinephrine will cause the dilation of systemic arterioles. I believe. Don't quote me on that. This is probably why I got that wrong. Mm. Okay. Anyways, and epinephrine will increase the cardiac output of the heart. Also a questionnaire. I love it. All right. So I'm going to talk about some examples or an example of baroreceptors being activated and some of the regulators that are important. Firstly, I want to describe the different uh, components of this reflex. So first we have a receptor. That receptor receives a signal, sends it through the afferent pathway, sends it to the control center, sends it back to the afferent pathway, and then receives an and activates an effector organ. So there's two different ways that this can happen. Like I said, the carotid sinus and the aortic arch. This kind of just goes through what I just talked about here. So an example with standing, uh, your your body is in kind of a not a fight or flight response, but a rest and digest. Therefore, there won't be as much excitation of these baroreceptors happening. The moment you stand up, though, a lot of the weight and gravity that um, on your heart and your lungs causes your heart to stretch a little bit, and everything else in your body to kind of stretch out, and that has the ability to activate those receptors and send a bunch of signals into your brain or into your heart. Sorry, thus um, but before you get dizzy because you don't have enough blood going to your head in a moment of you standing up, and then all of a sudden there's a ton of demand on your system. But then your that stretch more will cause an increase in heart rate and thus pump more blood to your brain. All right, so this has been um, Mr. Fizz. Catch me in my next video. Stay fizzy.